Okay, yeah.
My pleasure. Absolutely.
Okay, so Ariel, I'm just gonna present him quickly. He did his postdoc in computational neuroimaging with Brian Wendell at Stanford, where he actually met my previous PhD advisor, Michal Ben Shachar. And then he joined the University of Washington, where he started his own lab. His lab is mainly focused on developing computational tools to analyze diffusion MRI data and apply these tools to investigate a variety of research questions. And today, I think you're going to talk about the project of the cerebral pathways and schizophrenia, right?
Yes, I'll tell you about a few different things. I'm hoping to talk more generally about just our analysis approach and show you a couple of things related to human development, and then also talk about cerebellum. So I might, depending on how I do on time, I might skip through a few things, but I'll show you kind of the analysis approach and then a couple of different applications, including in cerebellum, since I know you're particularly interested in that part of the brain. So here, let me share my screen. Can you see, are you seeing now the slides?
Yeah, perfect.
Okay, great. So yeah, the analysis approach, we call it tractometry. It allows us to peer into white matter, so to speak. As Sivan mentioned, my group focuses on development of open source software. We develop open data resources. We focus on methods from statistics and machine learning and really on human neuroscience, a variety of human neuroscience questions. So generally I'm interested in detailed quantitative inferences about brain biology that can be useful to ask basic research questions, but I'm very interested also in thinking about clinical applications. And then the measurements we want to do are measurements that can be done in humans. And we think of the brain as a network. Sort of our basic analytic paradigm starts from the assumption that the brain is a network and that the connections between different parts of the brain are biological objects that we can analyze. And I'm intrigued by this idea of frictionless reproducibility, the idea that we will do something and then somebody else can take what we do and run with it. So I'll talk a little bit about that. So we develop open source software for science. Let me actually skip about that. So let me start by just stating what might be obvious, that the white matter matters. It's a big chunk of volume in the brain. You can see here on the top right that contains the axons of the neurons that connect between distant brain regions. And the health, the biology of the white matter is crucial for proper transmission of information between regions. In particular, the myelin, the thing that gives the white matter the white color, is this myelin that covers the axons, is crucial for proper timing of action potentials, for rapid transmission, and also for the reducing the energy cost of information transmission through the brain. And one of the things that's sort of one of the things we focus on is the fact that this changes over the lifetime. So it develops in early childhood, it develops in childhood and adolescence, and then in aging, it also sort of the myelination state of the brain deteriorates. So it plays an important role in all these fairly crucial points in life where a variety of things can go right, a variety of things can go wrong. But in addition to this kind of program that we have over the lifetime, we can also apparently it changes also in response to external stimulus. So this is kind of a classic study where people learned how to juggle, and you can measure changes, very specific changes in the white matter in the parietal cortex. So we can look at all kinds of things. We can look at sort of whether change is different in different individuals in childhood and adolescence, how that affects where they are, where they're adults, whether there are differences between different adult people and how that relates to individual differences in cognition. Then aging, we can look at resilience, how in some individuals these changes are slower than other individuals or maybe faster. So there's various places that we can look inside of this life course to ask a variety of interesting questions. And the tool that we use is diffusion-weighted MRI. Diffusion-weighted MRI relies on the fact that inside of these big bundles of axons that you see here on the left, there's water, and the water tends to diffuse more readily along the length of the bundles than across their boundaries. And we can tune the magnet to be sensitive to diffusion in a location and direction specific manner. And using that data, we can analyze that data in order to infer a variety of diffusion, what we call diffusion-derived tissue properties. So here's a horizontal slice through the kind of the middle of the brain, and you can see the ventricles have a lot of diffusivity, and regions of the white matter in the cortex have very low diffusivity because there's a lot of stuff in there that prevents the water from diffusing very far. We can also assess what we call fractional anisotropy, which is how much diffusion is preferential along some directions. And that tends to be very high inside of the core of the white matter. And then we can infer the principal diffusion direction as an inference about the primary kind of axon bundle in every voxel and its directionality. And using computational track tracing methods, we can take that information and track between different regions. So the way that we do that here, zooming on a horizontal slice, is we model the diffusion in every voxel, and then we put a seed somewhere, and we propagate what we call a streamline through the volume, following the directionality of diffusion in every location in the volume. And if we put a seed in a different location, we might go to a completely different place. So here we're crossing through the corpus callosum to the other side. Here maybe we're going to the posterior part of the cortex. And if we do this many, many times, we get something that looks like this, just a big bundle of spaghetti inside the brain, pictures you might have already seen. Now, tractography has all kinds of issues. There are inherent ambiguities of the diffusion signal. So let's say that we get imaging information that tells us, look, there's a crossing here, there's one fiber going here and another fiber going here. That could actually reflect many different kinds of states. It could reflect some curving inside of the voxel, or maybe there are two fibers that are kissing or crossing. And it's very hard to distinguish these. In addition, there's larger uncertainty and biases close to the cortex because there's partial volume with the cortex that's less, where the signal is less oriented. And if we rely on streamline counts, we're also subject to all kinds of biases that relate to shape, configuration of the streamline. So what can we do with tractography? Well, it turns out that brain connections derived from diffusion MRI tractography can be highly anatomically accurate if we know where white matter pathways start, where they end, and where they do not go. And that's the entire title of that paper by Kurt Schilling from 2020. So we rely on this principle that if we know where they're starting, where they're going, and where they don't go, we can derive what we call computational dissection, large anatomically valid white matter tracts. So here, for example, I've asked the algorithm to give me everything that goes through the corpus callosum. From the picture on the left, I've selected only the streamlines that pass through the corpus callosum and go to the anterior part of the cortex. And we can do that for many different anatomical structures. These are anatomical structures that have also been validated using other methods in anatomical studies, either in postmortem brains or in other species. So we know that they're real biological structures. And we focus on the quantification of tissue properties. Within these bundles, we do what we call automated fiber quantification. That means we take one of these structures, here, for example, the superior cerebellar peduncle, and we look at tissue properties in one position, and then another position, another position, another position, another position, and so on. And we get what we call a tract profile. So here's a tract profile of mean diffusivity along the superior cerebellar peduncle from posterior to anterior in the peduncle. And maybe we'll look at some other tissue property, maybe the FA in tandem, the fractional anisotropy in tandem. So we get these tract profiles, and we can use those to compare different individuals, to compare them across development or between different kinds of conditions. And we have open source software that we've developed called PyAFQ that implements the whole process. And we've developed around the software PyAFQ, we've developed a whole ecosystem of different software tools in collaboration with many collaborators that you see pictured here. That it starts from raw data. You can use tools such as QSI to pre-process the data, then run PyAFQ, and then on the output side, we have tools for visualization, tools for statistical analysis, and tools for machine learning and statistics. So it's kind of a big ecosystem of tools that we've developed over the years. So what can we measure with diffusion MRI? One analogy to think of is an analogy of a map that, well, if I'm sitting here in Kincaid Hall on the University of Washington campus in Seattle, and I want to get over to the Bill and Melinda Gates Center for Computer Science, there's this tiny little pathway, and maybe you can think of that like the single neuron axon going from one place to the other. And we can't really do that with diffusion MRI. And instead, we need to zoom out a lot. And instead, what we're looking at is we're looking at the highways. And though it's very interesting to study, of course, individual neurons and their properties and connectivity, knowing about the highways is also quite a useful information if you want to drive from one place to another in the brain or in Seattle. So that's the kind of thing that I think about is these brain highways. And we can ask all kinds of questions. I'll tell you a little bit about the kinds of questions we ask early in life and in childhood. We can ask questions like, is white matter development uniform in space? Is it contiguous in time? And what are the relationships between development of white matter pathways and development of gray matter pathways? And there are many more questions, but I'll show you some of the questions that we ask using these methods in childhood. So focusing on really the very early phase of childhood, really on birth. So in order to study that questions about development in birth, we use data from a large data set collected in the UK called the Developing Human Connectome Project. These are babies, infants that diffusion MRI data is collected in the first weeks of life. And there are a few challenges working with this data. Baby brains are smaller than grown-up brains, of course, but also their biophysics is quite different. They are much less myelinated than grown-up brains. And the white matter tracts themselves are different in terms of their shape, their trajectory. And so we collaborate with Marika Grotier, who's a world-renowned expert on this topic. And she's developed actually ways to track and delineate the tracts in infant brains. So here's an infant less than 2 weeks old, and we can see tracts like the corticospinal tract here, the anterior thalamic radiations, the cingulum, that are delineated in this brain. So we can delineate all the tracts that we can delineate in grown-up brains, or almost all of them in infant brains as well, thanks to Marika's work. So in this collaboration with Marika, we asked what happens at birth. So we can come up with a few different hypotheses about what happens in birth. We know that myelination occurs very, very rapidly and throughout the entire brain in utero, starting in the third trimester and maybe even earlier. And we know that myelination proceeds ex utero. And one hypothesis might be that birth is just a milestone along this way and development just continues in the same way. Another hypothesis you might have is that once the baby is born, they start being exposed to the environment and there's some kind of experience-based plasticity that increases the rate of myelination as soon as they're born. Or a third hypothesis that you might have is that they might have slower ex utero development, meaning maybe the environment is less protective. They have to learn how to feed themselves now that they're not connected through the umbilical cord. And so maybe there's some kind of slowing of development ex utero. So in this study, we wanted to adjudicate between these different hypotheses and looking at the Developing Human Connectome Project data. One kind of piece of terminology here is that we look at an individual baby, we can look at their post-menstrual age at the time that they're scanned, or we could look at the gestational age, which is the time from gestation until birth, or we can look at what we call the scan age, which is the time between birth and the time that they were scanned. And all of these time scales are important when we're talking about very, very young babies. Oh, by the way, here's a picture of one baby. This is the coil that they specially designed so that they can measure in these babies. So we're looking first at a cross-sectional sample. These are measurements from 300 infants. So this data set is rather large, which is very nice. These infants are born, the full gestational age at birth is 40 weeks. And some of these babies are born very early preterm. This is anything from 24.6 to a little bit delayed 42.3 weeks of gestational age. And they were scanned either, fairly early until, about 44 weeks. And that ranges between 0 to about 17 weeks of scan age. So the baby is measured very, very early in life. in order to quantify the myelination, we look at sort of simplified T1 weighted versus T2 weighted MRI ratio. Here is a T1 weighted in one of these babies. It's kind of unusual. If you've looked at T1 weighted images of grown-up brains, you'll notice that here the cortex is actually brighter than the white matter. And that's just the case here. And that's because their white matter is still very unmyelinated. Here's a T2 weighted, also odd looking if you've ever seen a T2 weighted in a grown-up. And here's the ratio. So you can see that, for example, the corticospinal tracts are at this point actually relatively very bright, really relatively highly myelinated. And so this is what it looks like. Here's two tracts, the frontal corpus callosum and the anterior and posterior corpus callosum. And what we can see if we compare three groups, very early, even pre-full term, kind of full term babies, and then babies relatively late after birth, we can see that there's extremely rapid change in the T1 weighted over T2 weighted, and this throughout the tract. It's actually true throughout the entire brain. So here are several other tracts, and we see this kind of very rapid change in this cross-sectional sample throughout the entire brain. So in order to ask what happens at birth, we designed two different statistical models. In the first model, that would be supporting hypothesis 1, we said, let's model T1 weighted or T2 weighted in every point along every tract as a function only of the post-menstrual age. So we're sort of ignoring when birth happened. We think only matters how long it's been since gestation. The other model breaks apart the post-menstrual age into gestational age at birth and then the age at scan. So this would differentiate between a model that thinks that there's only one rate of development versus a two-rate model. And what we find in doing a formal model comparison is that everywhere in the brain, this second model explains the data better. So we can rule out the constant development model. Now we can ask, given that this two-parameter model has two parameters, we can compare this and ask which is more rapid, the in utero, the parameter related to the in utero development that explains the variance due to gestational age, or the one due to scan age. And what we find is that pretty much everywhere in the brain, in utero development explains the variance much more than ex utero development. So our data supports this notion that there is slower ex utero development than in utero development. Let me actually pause here. I've been talking for a while and see what questions you have at this point.
Yeah, can I ask you a question on this?
Yeah.
Can you hear me okay?
I can hear you well, yes.
Okay. So I mean that's interesting, but doesn't it confuse two factors? So you could ask the question in two ways, right? For normal kids born around 40 weeks, what's the rate of development in utero and ex utero? That's one issue. And then the question is, if you have preterm babies, I mean, it's not clear that what they experience ex utero is, I mean, it's very different from what normal kids born at 40 weeks experience ex utero. They're going to be on preemie wards. Premature birth is definitely not something that nature has prepared us for, and so on and so on. So you could have you tried to dissociate the normal development from the preterm specific development? I guess that would be an interaction term in your.
Yeah, that's an excellent question, and we haven't done the full statistical model that also sort of classifies babies by this, but we have done the simple test of only running the model on babies born 37 weeks or older, which is 37 weeks is considered full term. Yeah. And we see this exact same thing there, but you're absolutely right that preterms are probably quite different than full term babies in many respects. And we in what I'll show next, we tried to dig into that a little bit more also into exactly because the sample is sort of enriched in preterm. We did try to dig into that a little bit more, but maybe our.
Yeah. But for the preterm babies, you would also find that they are, like if you measured them at the same age as the same gestational age as the real term baby, like the full term babies, you would find reduced wide matter development in them, likely, right?
Yeah, so that's a good setup for the next thing, because there is in this sample, there are babies, in particular preterm babies, that were measured twice longitudinally. One measurement done in preterm, sort of even before they reached full term age, and then another one that was done later on when they reached what would be considered full term. So we can actually design a kind of almost like a controlled experiment where every baby in this preterm sample that was measured twice is matched to a baby that was born full term, but they were both measured at exactly the same gestational age, or within very small difference. So we have a subset that's 34 infants that were born preterm and 34 infants that were born full term, measured at the same gestational age, so that we can compare and ask exactly the question: if you account for this, do you sort of account for any differences you find? So first of all, we find that there's a big difference between babies that were born preterm and babies that were born full term, and that even when comparing them exactly at the same gestational age. And so then the question becomes, is this fully explained, statistically at least, fully explained by the difference in gestation? So what we did there is we can take the model, the first measurement from these babies, and then we can apply the model that was fit on the cross-sectional sample. So this model was fit without these babies included in it. We can apply the model to them, and then we can predict the second measurement for these babies that were collected longitudinally. We have their first measurement done preterm, and then we apply the model to them. Each one we apply the gestational age that they experienced, and then the time that it passed between birth and scan age, and we can predict their second measurement and compare it to the true measurement, and that fits very, very well. So this model, this kind of predictive model, fits the data very, very well. So we can compare that to a constant development model that only takes into account the gestational age, and for every individual in our data set, the dual component model fits better than the constant development model. So in some sense, the in utero time accounts fully for the difference between babies that were born preterm and babies that were born full term. I think you were headed in that direction, right? So did that answer the question?
Yeah, thank you.
What other questions do people have about this?
So the decline that you see ex utero, does that correlate with the changing brain volume that's happening in the first few weeks of birth, and the increase or the pruning of the neurons? Do you have any way of looking at the changing brain volume at that?
No, not maybe. I haven't thought about it, but that's a great idea. I'm not sure if we can or can't, but I mean, because a lot of the data is cross-sectional, so do we? But in some sense, yes. The problem is that the measurements here were done up to like longest is 16 weeks post-birth, and most of them were actually done within the first 2 weeks of life. So but I guess we could. That's an interesting idea. We should look at that. That's an interesting one. Yeah. What we did ask, though, related to that, I guess, in some sense, is, and this is work that Steffi Zika, who's a graduate student working with Marika at Marburg University, is how is the development of the white matter tracts related to development of their cortical targets? So here's a few of these tracts, and some of these babies, or in one of these babies, this is a full-term baby measured not 2 weeks after they were born. We can identify based on the streamlines, we can identify for each tract what are the cortical targets, and we can assess T1 weighted over T2 weighted values both inside of the tract, but also at the cortical endpoints. And that's because the cortex is also developing very, very rapidly during this time. And what we find is a striking correlation between the T1 weighted over T2 weighted ratio in the white matter and the T1 over T2 weighted ratio in the gray matter targets. Here, the correlation is assessed across the different tracts. So each point here is the average value in the tract across all individuals in the sample. And what we see here is sort of this correlation across tracts. But we also see this kind of correlation within individuals. And we see basically that we can explain the change in white matter T1 over T2, or a lot of the variance in the T1 weighted over T2 over time is explained by change in the T1 weighted over T2 weighted in the gray matter targets of the individual tracts. So in some sense, the development of the tracts and the development of the gray matter is coordinated, which we find to be really interesting. It's as though they have kind of a shared plan, or there's some sort of shared biological substrate that is shared between the tract and its cortical targets.
A quick question there. So for the deltas, that makes sense. But if you did just T1 over T2 weighted ratio, these are slopes already in the figure. If you just did
these are already in the slopes. Yeah.
Oh, these are already slopes. Okay. So my question is just if you did just T1 over T2 ratio in adults in an adult cross-sectional brain, would you also find a correlation?
That is a good question that I actually don't know the answer.
Yeah, right. Because like for example, pyramidal tract should be highly myelinated and very clear, and then also intracortical myelinization in motor cortex is much more pronounced than prefrontal, for example.
Yeah, that's a really good question, whether this is a developmentally specific thing. Here, the change is so rapid that you can kind of see this change correlated with change.
Yeah, so
and some of the
I just wonder.
And that's very unique to. But I think that you're probably right that we would find a correlation over kind of tracts. This kind of correlation we might find in grown-ups as well. We do find that this, when assessed on an individual basis, also explains variance in language function much later in development, like in 18 months. So it's as though the coordination between the development of white matter and gray matter is important to establish sort of the ability to learn cognitive abilities, which is very interesting and intriguing in itself. Okay, let me just see the time. Yeah, I think I'll tell you more about brain development, just a little bit more. So, so now we looked at babies very early in life. The thing I'd like to show you is some work that we did on development a little bit later in life, in adolescence, and this using a very large data set called the Child Mind Institute Healthy Brain Network. It's a data set that has about 2,000 individuals in it, children between 5 and 21 years old. And we've done a lot of work. This paper from 2022 was work that we did to do quality control on the data, to process it, and so on. And we provided sort of an open data set with both the quality control, but also these tract profiles. So here you can see the development of the tract profiles here in FA, where each bin is a 3-year interval. So we go from darker green to brighter yellow in terms of age bins. And you can see that the development is not as dramatic as early life, but there's still kind of widespread development all over the brain. It's even more apparent when you look at the mean diffusivity. So here, mean diffusivity is assessed in every tract profile. And in order to understand the spatial patterns of this, Audrey Luo, who's a graduate student in Ted Satterthwaite's lab and with whom we've been collaborating on this, used a method to, again, use the simple statistical methods in order to assess the impact of development on individual positions in the tract. So here she's comparing two models. She's focused on mean diffusivity in this case, and she fits two models. One has sex, head motion, and sort of a within subject factor, and the other has another factor. So these two models differ only in the fact that one accounts for the age effect and one does not account for the age effect. This age effect is entered as a kind of a smoother, but so it's not just a linear. It's kind of like a smooth using a generalized additive model. And development here is quantified as a difference in the adjusted R squared between these models. And so what Audrey found is that development is not uniform in space between age 5 and 21. In fact, it's much more pronounced as you go further out towards the cortex than in the core white matter. So here are several subsegments of the corpus callosum. There are differences between the subsegments that are also interesting, but in all of them, you see this pattern that in the deep kind of core of the white matter, there's less age effects than when you go further out towards the cortex. And this is true both in the corpus callosum, but also in this set of projection tracts that you looked at. So this is interesting because it suggests that there is sort of some kind of developmental process that unfolds over time. I should mention Mareike, in previous work that she did, she showed that very early in life, we see the inverse of this pattern, where the core of the white matter develops more rapidly than the things that are closer to the cortex. So there might be a program that unfolds over time where early in life, the core of the white matter develops, and then later in childhood and adolescence, there's rapid development of the things that are further out, closer to the cortex. So this, I think, it complements nicely the work that we did with the babies and provides maybe a comprehensive, overall, we might have eventually a comprehensive model of development. I'm going to skip over this thing because of time, but it also, in Audrey's work as well, she has a preprint out, so you can read that one if you want. She also found that there's the coordination of the development of the tracts and development of the gray matter to which the tracts project, also in childhood and adolescence. So to conclude, white matter development is not contiguous in time, it's not uniform in space, and it develops in concert with gray matter, both in babies and in adolescents. Okay, so the moment you might have been waiting for is, okay, well, what about the cerebellum? So as we work on this, we talk to a lot of people, and we have a lot of conversations. Michal Sivan mentioned that I know Michal from my postdoc, and Jason Yateman, who I work with a lot, collaborated a lot with Michal on things related to reading. And one day, a few years ago, Michal sent us an email saying, hey, my student has worked really hard to delineate the tracts of the cerebellum. Could you include that in the software as well? And we got really excited by looking at the pictures. I'll show you some images in just a minute of these things, or maybe you've already seen images of these that Sivan's showing you. And started thinking, okay, what interesting things could we do with this? And at the time, I had a research student in my lab, Teresa Gomez, who doesn't like having her picture taken, so her picture is not here. And she was really interested in using these methods to study mental health disorders. And so we teamed up with Halil Aziz Velioglu and Heng Yi Kao, whose expertise is in studying individuals with schizophrenia. And part of the reason that we teamed up with them is this theory about schizophrenia and the role of the cerebellum in schizophrenia that some of you may be familiar with. This theory put forth by Andreessen and others is that there is a circuit, the cerebello-thalamocortical circuit, and that patients with schizophrenia have, or at least some of them have, dysfunctions in this circuit that may explain some of their cognitive coordination difficulties. And so we thought, great, let's find some data where we can look at this and examine the anatomical structures underlying this cerebello-thalamocortical circuits. I should mention Henge in his previous work, he showed in some data sets that functional activity differences through this circuit, the cerebellothalamocortical circuit. In particular, he found that there's sort of a hyperconnectivity throughout this circuit in clinical high-risk converters, more so than the clinical high-risk non-converters, even more so than the healthy controls in the samples that he looked at. So he said, great, let's use Sivan's work to find the anatomical substrate of the cerebellothalamocortical circuit. One component is, of course, the cerebellar peduncles, here shown in some anatomical images. So in particular, the superior cerebellar peduncles. And then the other component is the anterior thalamic radiation. And so we want to put together circuit both the anterior thalamic radiation and the cerebellar peduncles, and then evaluate the properties of those connections using our tractometry methods. So we looked actually at two data sets. One is a fairly old, openly available data set, the UCLA Consortium for Neuropsychiatric Phenomics, LA5C study that I'll just call CNP from now on, that has a large cohort, 270 some subjects, 49 out of which are diagnosed with schizophrenia, a large sample of controls. Their data is sort of fairly standard data for the time that this was collected almost 20 years ago. And because we have a large sample of healthy controls, we can match to every patient with schizophrenia, we can match a healthy control with similar not only age and sex, but also data quality control score, because we worry about the quality of the data being different between the different groups. And then in another data set that Halil and Henge collected at the Feinstein Institute of Medical Research, we have access to data from first episode individuals with schizophrenia, most of which are drug naive at the time that the data is collected. So these are very different. The CNP data set is chronically and very heavily medicated, actually, patients, and these are drug naive first episode subjects, so it's quite different samples. The MRI data is also different, much nicer MRI data, kind of state-of-the-art MRI data on this. And both data sets were processed using the same pipeline that I showed you before, including QSciPrep and also PyFQ. So here's an example of one of the brains. Typical for these kind of found data sets, you'll see that they were not particularly interested in the cerebellum, and they cut it off. So it's actually a tricky proposition to find the cerebellar peduncles, but nevertheless, we can find the cerebellar peduncles. And one of the hallmarks of the work that Sivan did is that these cerebellar peduncles actually decusate as they should. And so we can find the cerebellar peduncles. And here in the same individual, we can also find anterior thalamic radiation. So that puts together these two components of the circuit. And we can extract, as I showed you before, we can extract the tissue properties using PyFQ. In order to analyze the tissue properties along their length, we use generalized additive mixed models. The idea behind those is that we have a full tract profile, and it kind of wiggles up and down, and we'd like to compare that between different groups. In order to do that, we want to account for the different wiggles. So the first thing that we do is we model these wiggles as a linear combination of nonlinear functions. That's the generalized additive model. That's what it does. It models nonlinear or sort of smooth functions as a linear combination of nonlinear functions. In this case, these splines that are shown here in the middle. And so here is, for example, the combination of splines that accounts for this tract profile here in kind of the dashed line. In kind of like statistical formula notation, this means that we can fit a model where we have a group factor, that's the factor we're interested in. We're interested in testing differences between these groups. And then we have a smooth combination of nonlinear functions that accounts for the wiggliness, and we have a different smoother for every group. Then we have other factors like age, sex, which hemisphere we're measuring in, and the quality of the data that we insert into the model is an additional factor. And then we have this last thing here is kind of within subject random effect, or a random effect due to subjects. So let's look at one of these data sets, the CNP first, looking at mean diffusivity along the length of the tract. And what we find here is that the mean diffusivity is higher in control individuals than in individuals with schizophrenia. And this difference is statistically significant in the model that I just showed you. On the other hand, fractional anisotropy along the tract is not different between these groups. Similarly, in FE, we find a difference in the cerebellar peduncles, where controls have higher mean diffusivity and individuals with schizophrenia have lower mean diffusivity. Here we do find a difference also in fractional anisotropy between the groups. Looking to the next component, the anterior thalamic radiations, we find a small and significant difference where individuals with schizophrenia and the CNP have higher mean diffusivity in the anterior thalamic radiations, and they also have lower FA. In FE, on the other hand, we find sort of the opposite. We find higher mean diffusivity in the controls than individuals with schizophrenia. So, okay, this is a complicated set of results, hard to understand, and we're still trying to make sense of that. But at the very least, we can say we can make measurements of this anatomical substrate of the cerebellothalamic-cortical circuit in these two different data sets. And what we find is that the findings about the cerebellar hyperconnectivity in individuals with schizophrenia that are consistent across data sets, at least as we look at the superior cerebellar peduncles. And this is just to interpret this finding here, and that's the fact that the mean diffusivity is higher in controls than in individuals with schizophrenia. The mean diffusivity is usually associated with denser tissue. There are a lot of things that can account for mean diffusivity, but one of the things is denser tissue, maybe higher myelination, and so on. And then the findings about the thalamocortical connectivity are really mixed. And we think these might be related. If we have to interpret this, they might be related to differences between the data sets in terms of, for example, the medication exposure and just their really differences between these cohorts, primarily how chronic the schizophrenia is and then the medication exposure. So those are our findings about the cerebellum. And overall, taken together, these suggest that thalamocortical connections are affected by the chronic disease, while cerebellar hyperconnectivity may be more closely related to the common biological mechanism of schizophrenia.
Is there any postmortem work on superior cerebellar peduncle in schizophrenia, long-term schizophrenics?
I don't know.
Yeah, there is some work. There's a group in Korea that has done some work. It's hard to compare also because in a lot of the previous work, they don't have these nice decusating cerebellar peduncles. You find a lot of work, and Sivan can probably tell you about this, where people measure the intend to measure the superior cerebellar peduncles, but they measure these things that go ipsilateral instead of contralateral, which is just not the anatomical what we expect. So maybe you think, okay, well, maybe we can still measure tissue properties, and people have done that. And there's a whole mixture. It's very, one of the hard things about working in schizophrenia is that it's not one disease, and also there are extremely variable factors between cohorts related to things like how long they've been sick, the drug treatment, and so on. So it's really, really hard to tease apart different things. So that's been one of the lessons of trying to read this literature and interpret these results. Is it's really hard to study schizophrenia.
Are these differences that you observe in schizophrenia specific to the projections to the frontal cortex, or are they also in play for like the motor cortical projections and the thalamocortical projections that go to motor areas, for example? I might have missed that. I apologize.
Oh, that's a good question. So we haven't looked at other thalamocortical projections. We've looked at, and that's primarily because we don't actually have we have only two thalamocortical projections that we know how to delineate, which is the anterior thalamic radiations and the optic radiations, posterior thalamic radiations. So I actually don't know. I mean, this is work we could do to delineate. Oh, and I should say we now know how to do the acoustic radiations as well. So we haven't looked. We looked we looked a little bit at other tracts. We were always a little concerned about exposure to, well, about just kind of digging around in the data too much and p-hacking our way through. So we tend to come up with like a crisp hypothesis. This is where we should be looking because this is the hypothesis. So we don't tend to look a ton around these, but I've looked a little bit at other tracts, and there doesn't seem to be much there. I haven't tested it very thoroughly though.
Thanks.
I think you mentioned in one of the data set there was other disorders also. I was wondering if this change in diffusivity can also be seen for other disorders, or it's like it's specific to schizophrenia.
Yeah, that's a good question. Yeah, CNP has other disorders, and we don't find these differences. Spinocerebellar peduncle and the other disorders. Yeah, so it's among these three groups here. It's unique to the individuals with schizophrenia.
Thanks. I also was wondering if you had the chance to add the exposure to the drugs in a factor in the GLMs, and if that.
Yes, sorry, I should have mentioned that. Yes, yeah, we did. Yeah. This is I should mention. It's not on the slide, but it should be. I should mention that Teresa did. That was a lot of manual work to sort of figure out dosages based on information. The CMP does provide information about dosages and drugs, but you have to always do that relative to like haloperidol equivalent. So it's a lot of conversion that you have to do from one drug to another. So yes, we did this. Actually, the model does account for drug-related differences. We don't find a drug fact, like there's no drug effect by itself.
Thank you.
Yeah, cool. Okay.
So
I'll
this
just
all-
put up my thank you slide. Here, just to thank everyone again, all my collaborators and so on. Yes, and then I'd happy to take more questions about this or other.
So this is superior cerebellar peduncle that you looked at. What about middle cerebellar peduncle as the main input?
Yeah, so very hard in these data sets to find. It's harder to find overall. Maybe Sivan can tell me more about this actually, but it's actually really hard in this data set to find anything except the superior cerebellar peduncles because you start running into the limited slice prescription already.
Oh, because but otherwise middle cerebellar peduncle is bloody obvious. I mean it's big.
Yeah, we're working now with Hengi and Halil. We're working now on a lot more detail of the cerebellar connections in the human connectome project data set. There's this human connectome project focused on individuals with psychosis. There they collected real whole brain acquisitions, very nice data. And there we're working right now to delineate in a lot more detail connections of the cerebellum, including cerebellar peduncles, but also connections between, sort of subcortical nuclei in the cerebellum and the ventral tegmental area, and sort of other circuits of the cerebellum as well. But I don't have anything to show yet, unfortunately, because it's very tricky. Other questions about any of this? Thoughts?
Yeah, I mean it's in the cerebellar white matter, it's tricky. So I actually don't quite know. Maybe there's mossy fibers going through there, and then there's the output neurons of the Purkinje cells going through the white matter in the white matter body of the cerebellum. And
yeah,
I'm quite sure is in terms of numbers and myelinization diameters, which one would actually give you the diffusion tensor. And my gut guess is it would gotta be myelinated mossy fibers, just because of numbers.
I don't think we have the resolution, but Ariel, correct me if I'm wrong, to look actually into the white matter within the cerebellum. It's much easier to delineate the pathways that, the large pathways, the highways that connect the cerebellum to the rest of the brain. But within the cerebellum,
But
like
in the
partial
cerebellar
volume
white
effect
matter,
is
you
too.
have crossing fibers. You have the mossy fibers coming into the middle cerebellum. And then you have the Purkinje cells projecting down on the
Yeah.
on the deep cerebellar nuclei.
Yeah.
And they are crossing. They're gonna be at some angle.
Mhm.
My guess is that the diffusion tensor that you get in the cerebellar white matter will be dictated by the direction of the mossy fibers, just because of numbers. But I mean that is something that you should that should be. I mean that there should be some histological literature on that tells you what the hell we're seeing in the diffusion tensor there, right? And what we would see is retract tracing.
Maybe as I don't know, maybe in postpartum brains, it's easier to see that because you have greater spatial resolution. But I think that in like in vivo.
No, actually, like postmortem slice.
Yeah. Okay. Yeah.
I
Yeah.
mean,
Yeah, but I doubt that people have connected, right, between the. I'm sure there's histology of these things, but I'm not sure that anyone's ever connected the histology to the diffusion measurement. I think it's rather hard to do.
One thing we can certainly do is guesstimate how many fibers there are. So we know how many cells and how many roughly, right, up to plus minus a couple of million, right? And we know the diameters. And I think histologically you can see how big the myelin sheets are. So if like on the like, and then I don't know from those biophysical metrics whether you could on the back of the envelope do a calculation on guesstimate like what you're actually going to see. I mean the answer might be like, oh, you won't see Purkinje cells at all, right? In which case it's tracking output pathways from the cerebellar gray matter to the deep cerebellar nucleus is going to be completely hopeless, for example. Clearly, when you're like going from the deep cerebellar nucleus and the superior cerebellar peduncle, there isn't any that I know of any crossing thing going on. Once you're close enough to the axis, also inferior cerebellar peduncle when the spinal cerebellar comes in, right? Until you hit the white matter body, the mossy fibers, there's going to be very little fiber crossing. I mean, the center of the tract is going to be nice, clean mossy fibers coming from anterior spinal and posterior spinal. Right?
Yeah, I mean, I wish there was that we could say something like that, but honestly, I think at the resolution that we're working with, a lot of the variance is going to be explained by partial volume effects with just the cerebellar cortex, but also you might think that the variance between different voxels is explained by the prevalence of these different populations of axons and not necessarily primarily influenced by one population versus the other, making the interpretation really complicated. So I think we're in a really tough spot in terms of interpretation for sure. I think we can say fairly broad things using these methods for now, at least until we get to like really much higher resolutions, which will happen.
Yeah, I mean, not high enough to distinguish individual mossy fibers and Purkinje cell axons.
No.
I mean,
but yeah,
they're pretty hopelessly intermixed, right? In that.
Yeah, and to detect them with imaging methods.
Right, that's impossible. Yeah. Right, but I mean, at least like from histology, we can know how far they're intermixed, how many they are, and what the myelin sheets look like.
Yeah.
Right. So that I'm not quite sure that there ought to be somebody in the world needs to know, like, has some opinion. Looked at postmortem environmental tissue.
Yeah, I'm sure someone has opinions about it. But it goes back to my Google Maps kind of analogy also, where we just need to figure out the right questions to ask with this tool. I think it would be very hard for us to. It would be really nice to connect this to sort of the computational models of the circuit in the cerebellum, but I think we're pretty far from that.
Yeah. Cool.
Any other questions? Okay. Okay. Thank you so much, Ariel. Yeah, thank you for joining.
Thank you all. It was fun to
great.
present to you. Have a nice Friday.
That was great. Thank you.
Thank you.
Thank you.